Hello, everyone. I'd like to welcome you to our webinar on challenges of breeding tomatoes for both flavor and disease resistance. I'm your host, Alice Formiga of eOrganic. This presentation will last about 45 minutes, and then we'll have 30 minutes for questions. I would like to welcome back today Jim Myers of Oregon State University, Julie Dawson of the University of Wisconsin, and Jared Zystro of the Organic Seed Alliance. All of them are plant breeders who've been working on breeding tomatoes and other vegetable crops for many years, and they're all collaborators on the multi-state Tomato Organic Management and Improvement Project, also called the TOMI Project, funded by USDA NIFA OREI. And the web link on the screen allows you to find more information about the TOMI project. So with that, we have a lot to cover today, so I'm going to turn off my video and hand over the presentation to Jim Myers.
Well, thank you very much, Alice. I appreciate being here and being able to talk about tomato breeding. Um, the the TOMI project has been wonderful for supporting this collaborative effort. It involves Purdue University with Lori Hoagland being the principal investigator, Organic Seed Alliance, and University of Wisconsin, as you know from the people presenting here. North Carolina State, Virginia State University, University of Hawaii at Manoa are all involved in this project. Next, please. So the project has, well, this is actually the third iteration of the TOMI project. We're in TOMI 3, 4-year grants for this particular project. And in this iteration, we have 5 basic sections to the grant. One of these is on biocontrol, looking at beneficial microbials for disease control in various organic production systems. Second one, looking at plant microbial relationships, and in particular, looking at the phenomena of induced systemic resistance and how certain microbes can induce this resistance and how that might be useful in preventing disease in production systems. The third part is the breeding, which we'll talk about today, and I'll go into more detail in a minute on that. And then the fourth is developing decision support tools to support both breeding programs and growers and such. And then finally, there's an outreach and engagement portion, which is what we're doing today with this. So next, please. So the TOMI, the kind of the glue that holds the breeding section together are these breeding trials that are conducted across all the regions. We have trials in Virginia State University, North Carolina State University, University of Wisconsin, Purdue University. The Organic Seed Alliance trials are in California, and then mine are in Oregon here. These trials are replicated. We collect data on a small number of entries in these trials, and do things like look at yield, disease susceptibilities, flavor. Those are very important traits that we're looking at with these. And across region, we have some common entries, but then we may have region-specific varieties in some of the other trials. And that's partly driven because, and I'll allude to this a little bit later when we talk about disease resistance, that the diseases are different in different regions of the US. And you can see that in the Midwest and the Southeast, early blight and septorial leaf spot are primary concerns, while in the West, for us, late blight has been an issue along with powdery mildew. And the early blight is less of a problem. I don't think I've ever seen septorial leaf spot here in Oregon. So that's just the nature of the game here. Next, please. So just laying a little bit of groundwork here for talking about the various traits. Traits can be inherited or controlled by qualitative genes or quantitative genes. And we talk about these in terms of, For example, qualitative characteristics tend to be a discrete variation. It's either this or that. There's not a continuous variation. If you look at the graph on the right-hand side, that the top graph shows a qualitative distribution of you have one phenotype or the other. Sometimes you might see an intermediate in the heterozygote. They can generally be determined on a small number of individuals, sometimes just a single individual. It's relatively consistent across environments and generally controlled by one or maybe at most maybe three genes. Quantitative traits, as I mentioned, are continuously variable. You can see the bell curve in the graph at the lower right. And to measure these traits, you often have to have a large number of individuals, use replication, use an experimental design that allows statistical analysis, and you might have to repeat the test over seasons or over environments to get an accurate read on the genetic control of that variation. One thing in projects where we're doing mapping studies, quite often we can look at quantitative trait loci, and what we find are on chromosomes there will be a region associated with a particular trait, a phenotype of a particular trait, and that would be a quantitative trait locus. And so I'll use that term a little bit in going forward here. These quantitative traits are often controlled by a few to many genes. Okay, next, please. Here are examples of a couple of qualitative and quantitative traits in tomato. Fruit color is associated mainly with differences in different kinds of carotenoids. You know, they produce yellow, oranges, reds. The striping on the fruit, the skin color, whether it's transparent or whether there's actually color in it, those are all qualitative genes that control that aspect of the color of the fruit that you see. Quantitative genes, in this example, would be the fruit size and shape. There's a major QTL for size, but then there are many other genes that influence that size, and then a number of genes that will influence the actual shape of these traits as well. Okay, next, please. Some of the key diseases in tomatoes. This is by no means an exhaustive list, because if you go to other regions, you may find that there are some that are important there that maybe, particularly in the tropics, that we don't necessarily see here. I've also left off some of the popsiviroids and some of these new viruses that we're beginning to see now. But anyway, the fungal pathogen, bacterial, well, the fungal and viral and nematode pathogens I've listed in bold here are quite often what you'll see on a seed packet. You see the abbreviations VFN or VFTN. These stand for Fusarium wilt, verticillium wilt, tomato mosaic virus, and root knot nematode resistance. Some of these others are important and are being incorporated into varieties. Tomato spotted wilt is one that many commercial breeders are incorporating. In the Southeast, bacterial spot resistance is one that's very important. Such as septorial leaf spot resistance is important. Leaf mold is an important disease in high tunnel production in general. So next, please. Disease, and I've mentioned this, diseases vary by climate. In the U.S., one of the major differences is and separation, geographical separations you see is with the continental divide. Humidity tends to be higher to the east of the continental divide and much lower to the west. And the east, the humidity favors bacterial and fungal diseases. In the west, the arid climate tends to favor virus diseases and certain diseases that thrive under arid conditions. in particular with viruses in the West, it's aphid populations that may be more important than the actual presence of the virus. Um, and in addition, northern latitudes, we tend to see fewer diseases. Southern latitudes, we tend to see more diseases. And that generally uh, uh, that controls pretty much where things are shown. Some of these we see, throughout the U.S. early blight, tomato spotted wilt virus, but they tend to be more important or more severe in some others than others. Okay, next, please. One of the things about tomatoes and is that we have uh, gotten a lot of our traits out of wild species, wild relatives of tomatoes. And this particular species of tomato, our slide here shows some of the different species of tomatoes and their relationships to cultivated tomatoes. So the series Lycopersicum has the cultivated tomato, Solanum lycopersicum, but also some others like Solanum pimpinellifolium. You might know that as a current tomato. And then a couple of species from the Galapagos Island, Solanum cheesmanii, and then one I don't have listed here called Solanum galapagos. Anyway, those are the closest relatives to the cultivated tomato and generally easy to cross. Those are what I would call the colored fruited types. They ripen to a red or an orange or a yellow color. And then the others, the Neolycopersicum and the area persicum are more distantly related species, and these don't develop the carotenoid colors in the fruit during ripening. Most of those can be crossed to tomato, but cultivated tomato, but they may require special techniques like embryo rescue to be able to transfer traits. One of the consequences of having used all these wild species in the development of tomatoes is that modern cultivars actually have more genetic diversity than land races. And we think that's a consequence of crosses to wild species. You may be selecting one particular trait, but you'll find on a chromosome, you're actually bringing in a segment of a chromosome, and there may be several genes in that segment, which increases the genetic diversity. Next slide, please. This is a rather busy table here that shows where some of the sources of resistance genes have come from that we use. And you can see, for example, with rudnon nematodes, this has come out of Solanum peruvianum. Solanum pimpinellifolium has been a source for many resistance genes like Fusarium wilt, race1, tomato, well, let's see, many others here. The late blight resistance genes, the leaf mold, verticillium wilt, and so forth. Some other sources, in addition, have been Solanum pinellii, Solanum chilense, Solanum habercites as sources of resistance. This slide also shows you whether the genes are qualitative. These would be listed as the R genes, or QTLs, which would be more of the quantitative resistance. Next slide, please. One of the issues that one faces when integrating these genes and working with them is that sometimes they're located close together on the chromosome. This slide shows a table with the chromosome numbers at the very left, the locus for a particular resistance, and then on the right is the approximate position on the chromosome in megabases. And the cases where I've circled it in yellow, these are where genes are adjacent to one another, or maybe even in the same gene cluster. And in those cases, if you have, say, resistance to one linked to susceptibility to the other, and you're trying to cross that to a parent that has just the opposite, it can be quite difficult to combine those because there's tight linkage there, and you have to look at very large progenies to break that linkage. So next slide. In addition, I mentioned that we're introgressing from wild species, and in some cases, when we bring in these larger chromosome segments, we're also bringing in deleterious genes that you can see some of the known deleterious linkages that have been reported for some of these various genes. Things like reducing fruit size, increasing sensitivity to another disease, bacterial spot in the case of the I3 gene locus for Fusarium wilt resistance 3. Various, yield drag may be a problem with some of these. So one of the things that can be done is if you can find map the location of these genes and eliminate some of this, the wild germplasm in those segments surrounding the gene, then you can eliminate these this linkage drag that may occur. Next slide. One of the things in terms of screening for resistance to diseases is how you go about doing this. You can set it up in some cases in the greenhouse if you can grow up the pathogen and do your inoculations there, or you can do it in the field. And then the alternative to that is to marker assisted selection, then with validation at the end using field screening. Field screening and greenhouse screening tends to be more labor intensive, uses lots of resources. It may use a lot of time. You have to consider things like plot size. You may need replicated plots. You may have to inoculate, or ideally you may have a system where you have inoculum in the present that comes in naturally that you can use. There may be questions about what race of organism that you want to inoculate with. Some maybe, if you're screening for Fusarium race 3, then you're going to have that race present when you're screening for that particular resistance gene. The picture here shows a field where we have late blight coming in. You can see it's very clear in this slide that what is susceptible to late blight and what is resistant. But in some cases, you may need quantitative scales for rating your material as well, because the differences may be just very incremental. Next slide. So some of the work we've done with late blight using marker-assisted selection, this is finding markers that are located close to the resistance loci that you can use then to select for resistance. We have markers for pH2, pH3, and pH5 for late blight. pH2 and pH3 are commercially in some cultivated tomatoes that are sold commercially. We've been working with pH5 because this adds another layer of resistance to this, and it's not been yet deployed commercially. But you can see that you can pick out the resistant versus the susceptible genotypes based on the marker, the movement of the markers on these gels, and you can also pick out the heterozygotes as well. Based on these, then you can select individuals that are resistant to all three of these, carry all three resistance genes, and then test these in the field for further to validate that you do have the resistance present. So next slide. And this just is out of some of Julie Dawson's work, graduate student Amber Carvalho, with showing the validation of the septoria resistance. There are two different loci here and you can see the resistance, the combination of the resistance alleles on the left, the heterozygote in the middle, and then the susceptible alleles on the right. And with that, I'm going to turn it over to Julie.
Great, thank you.
Hey, Julie, just click on the screen there and you should have the control.
Yeah, thank you. Thanks, Jim, for that great overview of how complicated some of these disease resistances are. I am going to go through some of the challenges in combining flavor and disease resistance, and how it is often very challenging to get both in a cultivar that works well in organic systems for local markets. And then Jared is going to walk through some of the nuts and bolts of how we are doing this in the TOMI project. This graphic shows the results of a survey that my group in OSA did to look at what farmers wanted to know about new varieties before they would try them. And flavor was the top for tomatoes. Disease resistance is a very close second, and then yield, and then earliness, days to maturity. And so this combination of flavor and disease resistance is critical. It is what farmers need the most, and it is very challenging to actually accomplish. I also want to start with the acknowledgements. So what I'm going to present today is largely the work of Ambar, who I think is actually on the participant list. So if we get questions I can't answer, then she gets to do that. So Ambar did her PhD at UW-Madison and is now Dr. Carvalho and working in British Columbia on participatory plant breeding. And then Marissa Nix and Toppin Sickley, I think I saw Marissa on the call as well. And we worked also with SeedLinked, Nico and Gelbert, who's there in the photo too. Obviously funding came from TOMI, and then a few other projects complemented that. So the trade-offs I'm going to talk about with some data today are from a crossing block that was largely designed to start with good flavor. And you do not need to remember what any of these are. The main purpose here is that our parents were selected first because they were performing well in organic trials for flavor and had reasonable disease resistance. We did not have major markers for septoria or early blight, but we had some level of tolerance to those diseases. And septoria and early blight are really the critical diseases in the Midwest. And so we started with this crossing block to try to develop varieties that worked well in organic systems and that had very good flavor with acceptable disease resistance, with the idea that we would continue to improve the disease resistance, but we didn't want to start with something that didn't have good flavor because it's often harder to fix flavor in varieties. The first couple of years we were in the field, and then we quickly realized with this group of crosses, we really didn't have strong enough resistance to septoria. However, the high tunnel environment actually allowed us to move forward with these because that reduces disease incidence a lot. But even in that case, I'm going to show you some trade-offs between disease resistance and quality. The next steps, which I'll talk about at the very end, include bringing in that stronger septoria resistance so that we can test these in the field as well with hopefully the stronger disease resistance. We did look at yield, a lot of different production traits, as well as other diseases. Septoria was the big one, but we looked at a bunch of different diseases. And then we evaluated quality, both in the lab through BRICS, which is sugars, and then citric acid, which is what Ca is, and that is a measure of the acidity. And we did tastings with our crew. Some of these tastings started during COVID, so we all had little samples. And then later we were out in the field and doing this as a crew. We really tried to standardize the methods for the crew as much as possible with a training so that people would be scoring them similarly, and trying to get through all of the lines in the trial so that everything did get rated. And that is possible to do with a crew because they're here all summer. They are usually pretty excited about tasting tomatoes, at least at the beginning. And we can get consistent results across tastings. And Marissa has been really instrumental in setting up our protocols for tasting. We also wanted to get more of the user data on flavor and on performance. We had farmers participate in the selection process. We had 6 in 2020, 5 in 2021, and then we used SeedLink to really get feedback from a much broader range of farmers on the lines that were doing well. And this included a holistic assessment of flavor, not ratings for everything from sweetness to acidity to bitterness and umami, but a holistic rating of flavor about whether they thought that was good or not. And then we worked with culinary professionals to really do a more thorough evaluation of characteristics, but only on a very limited number of samples. So this is kind of the first trade-off, is you can get very basic data on a lot of samples, although you're still limited by the number of tomatoes that people can taste before they go crazy. But you can only get detailed data on a much more limited number of varieties. And so you want to make sure the varieties getting there are the ones that are most promising, that have a good combination of both disease resistance and flavor. And that, I think, is the real challenge, because we have kind of this general phenomenon in plant breeding. It's a numbers game. The bigger the population you have, the more likely you are to be able to actually find the combination that you want. But every time you add another trait to that combination, your population size that you need goes up exponentially. And so we do have a lot of traits we're selecting for in tomato. We have all the agronomic traits, we have multiple disease resistances, and we have flavor, and that makes it challenging. So with the CREW data and the lab data, we were able to look at correlations. We had enough data on all of the different lines that we were able to look at correlations between some of the disease resistances and the agronomics and the quality traits, to see kind of how much of a negative correlation we might be dealing with. And I think we were pleasantly surprised that it wasn't as strong as we were fearing. We had, of course, some lines. So how you read this is this, as it goes up, it gets worse in terms of disease. As it goes in this direction across the x-axis, you get higher yields. And as the color gets more red, you have more sugars in the tomato fruit. And so we have this variety here that is not as good for disease resistance, but is reasonably productive and has very high sugar. We also have some, like Defiance, that's our check. It has good disease resistance, very high yields, but only moderate sugar. We have some in here that are kind of very low sugar, but reasonable resistance. The one that is down here has a pretty good disease score. The marketable weight may not be great, but the sugar is pretty high. And so we had some that we can actually pull out of this that are better than what you would expect if you had a very strong negative correlation between the sugar content and the disease resistance. And I think in the conversion to this slide format, we lost the citation, but I will put that in the chat because this is published so people can go look at it if they want more detail. The numbers and letters, I will show you a little more information later on these breeding lines. This is a different way of looking at the correlation, which includes a lot of the agronomic traits, so marketable by count, average weight, how much is unmarketable. Then we have the lab data here, which is the Brix and citric acid. And then these ones at the bottom are the ones that we have the CREW actually score: appearance, texture, sweetness, acidity, bitterness, umami, intensity, and the overall score. And then the correlation goes from strongly negative at -1, which is red on this scale, and then 1 is very positive, and that's blue. And so you can see some of the flavor traits are highly correlated positively with each other. So intensity and overall rating of flavor are very highly correlated. But the overall flavor is slightly negatively correlated with the yield, for example, and with how big the fruit is, which is not unexpected. And so the takeaway from this is, one, we need to be able to evaluate large population sizes to find those individuals that combine both disease resistance and flavor and productivity. We also need to choose parents very carefully. So being able to integrate stronger disease resistance through marker-assisted selection is going to be useful so that we can select mostly for the background that is good flavor, but with the disease resistance trait in there. And then making sure that we have these professionals involved at the end so that when we have selected kind of our best combination, we can see how it's actually going to do in the field. And this is a point where I just want to make a comparison in terms of vegetable breeding programs versus some of the grains breeding programs, because often the population sizes is largely dependent on how much it costs to phenotype something. And disease resistance in the field is very expensive, as Jim said. It can be unreliable if you don't necessarily have the right inoculant or you have a season that's not conducive to disease resistance. And so measuring yield and disease is very expensive in the field. Measuring flavor is actually a little easier, although it's still pretty expensive, and it takes a lot of volunteers or time. The lab measurements are pretty straightforward, and we've been able to do a lot of those. And then we've been able to develop some methods for flavor evaluation that get through quite a lot of lines, although it's not hundreds or thousands. In contrast, for something like grains, field evaluation can be fairly straightforward. Disease is still a little tricky, but the quality evaluation is much more expensive. And so there, you kind of have the opposite situation where it's actually more expensive to do the quality evaluation than it is to do the field. I think in the tomato case, the fact that the disease screening is very expensive and we do have some good markers means that using those markers in order to bring that disease resistance into things that taste good already is going to be a good strategy. And so just kind of as the process moves, we started with these crosses. We used the crew tastings because we could do more in order to actually get to varieties that we were able to send out to farmers and to do seed chef tastings. Once we had that crew data, we used that to essentially bring the best lines to the chefs, combined with agronomic data. And we're at the point where, starting with all of these lines, we're down to 2 that we want to potentially release, and then another couple that are useful for parents. I'm not going to go into these in depth, but they are very good for some of the agronomics and the flavor. And the asterisk is that they still do not have strong enough disease resistance for field production. They are very good for high tunnel production. Same thing with the parents, for the varieties that we're not looking to release as varieties, but for looking at parents. Also really good productivity and flavor, but still working on that disease resistance for the field. So I think there are 2 strategies really to do that. One is looking at hybrids, and some of the seed companies that we're collaborating with, including Frog's Loop Farm, are looking at making hybrids with disease resistance as one parent, like a parent that is very good disease resistance, and then the flavor parent as the other one. Often this is a strategy. I've put a few varieties on this slide that kind of use that hybrid strategy to combine really good flavor with good disease resistance. Hybrids are a faster way often to get to that than trying to breed an open pollinated variety. And so these are ones that you might see on the market sooner, where we have with some of the independent seed companies working with these sources of disease resistance, they might have good disease resistance and good flavor in a hybrid variety soon. And then the other piece of this is looking at how do you get homozygous disease resistance for multiple resistance loci for septoria or early blight or others. How do you get to this plant that is both resistant and looking very good in the field and has good flavor? And so we're using markers in order to do that. We're in kind of the 2nd generation of making crosses and hoping to incorporate some of these better disease resistances into varieties like the ones I just talked about that have been selected for pretty good tolerance for diseases, but not that strong resistance that you need in the field. So with that, I will turn it over to Jared.
All right, thank you, Julie. So we've gotten to hear a little bit of some of the context and background and challenges around breeding for both flavor and disease resistance in tomato. And I just want to talk a little bit about some of the specific ways in which those challenges have manifested for us in the TOMI project and some of the strategies that we've been employing to be able to incorporate both of those goals as well as other breeding goals in this breeding program. So first thing talk about is kind of the disease resistance piece. So Jim introduced the concept that some disease resistances are qualitative or can be controlled by sort of single alleles, single genes here, and some are more quantitative where there might be multiple alleles interacting. And what we started with in TOMI is a bit of both. So some of the parents, so for example, Crimson Sprinter that was apparent in some of these earlier iterations there's been some evidence of it having quantitative septoria leaf spot resistance. But then in addition, when we started looking at and considering parents to include in crosses, started thinking about what genes were they known to carry. So this is kind of a grid of them, some of them carrying it as homozygotes, and then some with the hybrids carrying them in a heterozygous form where it's only coming from one of the two parents. So we kind of, similar to what Julie had mentioned in the UW program, had done a crossing block to incorporate these diseases, seeing if we could stack them as best as possible in ways where the parents, the kind of the disease package from each of the parents would complement each other. Oops. The other challenge that exists in breeding for disease resistance that you've heard already is the inconsistency in both. These diseases are regional, but they're also not necessarily always present in each region each year, or in even a region where they might be an issue in some years. They're not an issue every year, or they may not be an issue either. The severity or the timing might be such that it's not really something that we can use very well for selection, or they may be so severe that we're not able to see differences. Now, one of the great advantages and strengths of this project is the fact that we do have sites scattered throughout the country. So between California, Washington, Oregon, Indiana, Virginia, Wisconsin, North Carolina, we're able to see diseases that are only showing up in some of those regions each year, and be able to have, even though we are doing a lot of individual breeding for our programs, there's kind of an opportunity to use each other's locations as disease screening opportunities. So it's kind of an example, this showing an evaluation that we did for some of the diseases that we are seeing. And what you can kind of see here in this slide is that not all of the diseases were present in each location. So kind of as an example here, this year, when this data was recorded, the early blight was only showing up in Purdue, where something like the late blight down there at the bottom, we saw in multiple sites. But even I think what's helpful to see is even within, if we look down there at the bottom, within the multiple sites where late blight was seen, at some of the sites, like I think at Oregon here, which was which is blue, the blue dots there all the way at the bottom, the severity of the late blight that was seen and recorded maybe wasn't severe enough to really differentiate between the different breeding lines. Whereas, over in North Carolina and I see, actually it was NCAT was the blue that we didn't see differences. But in North Carolina and Oregon, we can see not only was it present, but there was enough severity that we can see this spread where some of the more susceptible lines are showing up as different than the more resistant lines. And so by being able to have this great trial network that exists within Tomy, it's an opportunity for us to be able to look at more diseases, and also a little more likely that we're going to see one of the diseases that we care about selecting for, hopefully with enough severity to allow us to make good selections, and at least be able to see that disease at least one of our sites each year. The other thing that was mentioned before, and something that we've employed in Tomy, is taking advantage of the fact that we do have some of these single qualitative major resistance genes that we can be using marker-assisted selection for. So some of, and I think Jim sort of talked about this a little bit, for some of these diseases, the markers are still being developed. This is always kind of a first step, is a disease may be a qualitative disease. There may be recognition that this is controlled by a single major gene, but until you can find a marker that you can select for in the lab, you're not able to do the marker-assisted selection. So once those markers are developed, there are a lot of, and this is something for all of you out there, there are commercial services, including one that we used, that will allow you essentially to take, you can kind of see on the left-hand side there, or you can take leaf punches of plants. So this is an opportunity to actually evaluate on a plant-by-plant basis. You can take leaf punches of those plants and put them in a well, like a 96 or well tray, and send them off to commercial services for a relatively reasonable price. They can tell you, what did we see? Was it resistant? That's what the R is. Was it a heterozygote? That's the ST. Or was it susceptible? Was it that's the S? And so in our case, we were able to make our crosses, knowing that we had often some of these major resistance alleles coming from one of the two parents. And then in the F2, if you recall back to Julie's sort of breeding funnel slide, this is where you make the initial cross. That's your F1 hybrid. That generation after that is known as the F2 generation, where you have kind of the maximum diversity. We can screen in that F2 stage or later, and identify which of these resistance genes is present in these different individual plants. And then once we've identified which of those plants have the genes that we're interested in, we can save seed directly from that plant. And this is something, thanks to Joel and others at Oregon State University, we're able to do in the winter, in the off season. And so this was something that could kind of augment our field testing and kind of speed up, bring on an extra generation of advancement combined with this marker-assisted selection. Of course, all of the kind of caveats I think that Julie started raising around, we're, by doing the selection strictly in the lab, we are not capturing the rest of the important traits here. We're only looking at whether or not these disease-resistant genes are present. And so this would not be something that you would want to employ in a vacuum, right? This is something that you would want to combine still with that field testing, both for those quantitative resistance genes that aren't captured by this marker-assisted selection, as well as for the the agronomic and quality traits that you're going to need to be handling real plants and fruits to be able to make judgments on. One of the other, so for flavor evaluations in Tomy, we have often streamlined it down to kind of a single, I guess what you call hedonic rating, a single rating of overall flavor. And one of the challenges that we face in any taste assessment, I think Julie brought some of this up, is just that everyone's, even it's first off, even with a trained panel, people's preferences differ, and their scores differ. And so you can see kind of like that picture on the right there of, if you have just everybody, this is people just rating an individual variety from, 1 to 10 or 1 to 9 on a overall flavor scale. You can see just a huge spread of ratings. for a given variety. And the question is, well, how can that information be useful? So one way that you can make it slightly more useful is to tease it out by blocking by taster. And what I mean by that is that often not every taster is tasting every fruit, but if you have a way to associate each rating not just with the variety, but with an identity for who did the rating, now you can kind of pull out this this taster effect. So if there's someone that just kind of across the board gives everything lower ratings whenever they rate it, that doesn't, it helps reduce the risk that they're sort of skewing the ratings down for everything that they did taste relative to the things they didn't taste. And so you can kind of create somewhat of a balanced structure where even if not everybody's tasting everything, people are tasting the same amount, and that each variety is getting tasted by the same amount of people. And then you can pull that kind of taster effect out to some extent to give a little better apples to apples comparison between the different tasters. Relatedly, and I see this popping up in chat, one of the other challenges that we face, well, there's two. There's also a variation, and I see this mentioned, like kind of fruit to fruit variation within a given plant. So there are some strategies around this. First off, and we didn't get into the nitty gritty of it. I wish I had a picture of it, but standard practice with tomato tasting is that even within a single fruit, there's variation in flavor in the different tissues in that fruit, right? The skin versus the flesh versus more of the juice and the seeds and the gel sacs around the seeds. All of these have different flavors, and so if you're not eating a whole tomato, you want to think about having a slice of that tomato that is representative of the whole, so more of a wedge, right, rather than cubes. So that way each bite you're tasting kind of all the different tissues in relative proportion to their distribution in the fruit. And then and then taste, if at all possible, multiple fruits from that same plant, because whoever was writing that in the chat is right, there can be variation just fruit to fruit within a plant. The final thing that we've come across quite a bit, and I know Julie's looked at this a lot, is that there can be a pretty wide shifts in perceived flavor based on date, and in particular based on kind of environmental influences that are happening throughout the season. So this can include, was there a rain event, just prior to tasting? How much disease pressure is occurring at different points in the season? This can affect the flavor. Just temperatures can affect flavor. And so another strategy here is kind of similar to the way that you can try to, if you can't evaluate every single variety on the same day, trying to create that similar sort of block structure where you say, okay, we're going to evaluate these ones all today, these ones all next week, and we can create this date effect. You can start to pull a date effect out, so that way if you tasted everything next week and it was all after a rain, if there's an overall decrease in the flavor because everything's a little more watery, you can try and adjust for that. So those are some of the strategies that we've attempted in the TOMI program to balance the disease resistance and flavor selection using this whole team that we have, and using both the tools that we can do in the field and then in the lab. I think that's all. I think that was my last slide. So I think we can switch to answering all these questions that are starting to appear in chat.
I just wanted to thank you for the presentation, and then also to say we're going to get on to the Q&A in just a second. But for anyone who missed the very beginning of the presentation, we did record it, and you'll be able to find the recording on the eOrganic YouTube channel within the coming week. I know before the webinar we got one question, or we got actually two questions by Glenn, so I wanted to make sure we cover those. You had definitely talked about some of the diseases that we should be most concerned about, but are there any seed-borne diseases that we should be most concerned about? that are affecting tomatoes right now?
I guess I can start out with an answer on that. So the bacterial diseases can be serious seed-borne problems. Bacterial spot, bacterial speck in particular. Some of the virus diseases are an issue. These are some of the new ones, the popsiviroids I mentioned at the very beginning. These are potential problems. Brown rugose fruit virus, which is a fairly new one, I think has a disease component or a seed-borne component as well. There are differences in some of these diseases. Some of these diseases will go internally. The pathogen will be internal to the seed, and those are probably your most serious, whereas some of the others are external, but kind of difficult to remove. And there are various processes you can go through in uh, taking your seed, putting it through like a hydrochloric acid uh, uh, digestion to remove the pulp from the seed, but also it will kill a lot of the pathogens that might be external. Jared or Julie, do you have anything to add to that?
Yeah, I pasted in chat here. I think this came from University of Wisconsin Extension, but there are some great tables and tools out there for using hot water treatment to control at least those superficially seed-borne diseases. There are both some good protocols there. It certainly will reduce your seed kind of longevity so that you're typically, doing this kind of treatment just prior to planting. It's gotten easier, I would say, nowadays because you can purchase, there's some good protocols for just using a relatively inexpensive sous vide cooker to obtain these precise temperatures, whereas previously, it was more limited to needing like a more laboratory hot water bath. So I feel like this hot water treatment is becoming more accessible to more people.
Okay, we have a couple questions from Roland here in the chat. He was talking about the variation at different times of the day, and I know you already talked about that in terms of the Brix rating. But he also asked, what is your experience with hybrids and the Brix in F1? So for example, well, he kind of gave an example. I don't quite understand, but it's Brix 6 times Brix 6 gives what, or Brix? Yeah, so anybody want to tackle that question?
But just comparing hybrids and heirlooms, I think in general, the perception is that hybrids have worse flavor and heirlooms have better flavor. But we found just as much variation in heirlooms as in some of the hybrids. And you can find modern varieties, either pure lines or hybrids, that have good flavor. And you can also find heirlooms that have good flavor, and you can find bad flavor in both categories. In terms of the expectations from a cross, I think it depends somewhat on if the parents are actually, combinations of grandparents that had contrasting flavors, or whether the grandparents had more of similar performance. Because if you take an F1 and you self it, the amount of variation you're going to get is going to be partly a function of like how variable were the grandparents that you crossed. And you don't always know that for commercial F1s. I don't know if that answers the question or not, but you can get quite a bit of variation sometime in an F2, and you can get not so much variation sometimes in an F2.
And I would just add one thing with that Julie alluded to, which was the size can influence the Brix. And that's pretty well documented that smaller fruited tomatoes generally have higher Brix. And so if you're making a cross between a small versus a large, I think that might influence what you would see in terms of the Brix. Probably something intermediate between the two.
Right. And in
Okay.
general, that is a quantitative trait, which would mean that you are expecting to see kind of a normal curve where you have, most of the things intermediate to the parents, and then you may have few individuals that are either worse or better than both parents, rather than having a population where you have classes of, high Brix and low Brix. And Jim, you've done a lot more of this than I have in terms of evaluating progeny families, so feel free to comment as well.
Okay.
The other thing about evaluating Brix that no one's mentioned is temperature makes a difference. And like if you're trying to taste tomatoes, not just Brix, but also flavor, at the end of the season, you often can't really get very good results after you've had some cold weather, and the sugars and aromatic compounds just aren't being created in the fruit once you have some cold temperatures. So that's another factor.
Yeah, and cold temperatures also affect the texture. So it sometimes is very hard to separate that towards the end of the season, because nothing is tasting great, partly because they've been below 50 degrees maybe at night, and that is affecting the texture. We do try to do this at the same time of day. It is sometimes easier than other times to actually hit the same time of day, but we try to do this if we're doing individual kind of plants with a smaller group. We'll kind of block out 2 hours each day at the same time to do it. And if we're doing it with our crew, it would be the same time each week, where we're tasting things with a larger group of people, but it wouldn't be every plant in the population. It would be kind of a bulk sample.
I have a related question for you two. Thinking about the temperature and flavor. How if either of you tried evaluating kind of for flavor holding potential? I know, like for example, just I've spent more time in the world of sweet corn, and the taking a bite test in the field is one thing, but right, most people are going to, it's going to be picked, it's going to be put on ice, it's going to go to the farmer's market or the store, it's going to be a couple days before people bring it. So we would actually sometimes evaluate by harvesting, putting something in the cold room for a couple of days, then doing our taste test to have a better reflection of what consumers would actually be experiencing. Have either of you tried anything like that with tomatoes? And if so, have you found any differences in terms of how well tomatoes hold their flavor?
No, I've not looked at anything like that, Jared. It'd be a complicated trial to set up. It could be done, but I don't know. Julie?
Yeah, same here. I think what we try to do is only taste and measure Brix on the ones that are ripe, which are not the ones that would be harvested in a longer, kind of chain system. Farmers, however, do harvest slightly underripe to take things to the farmer's market, because otherwise too much of it splits. And so what we've been really focusing on is the flavor potential at full ripeness and the Brix potential at full ripeness. The farmer would probably pick a little earlier and then ripen it, on the way to market. In a supermarket, you're picking it really early and then often using ethylene or something to ripen it to get to the grocery store. In that situation, flavor is not the priority. So I think for our market, because of what we are focusing on, we've really tried to compare everything at fully ripe stages. I think we could do the kind of pick them when a market farmer would pick them and hold them about 55 to 60 degrees and then taste them then. The biggest barrier to that is just the time it takes, because it's a trade-off between doing more evaluations in the field, so we can get more lines and potentially more combinations, and doing multiple evaluations of each line. And that's, there's not a right answer to that, but we have gone towards more lines and not multiple tasting, parameters.
Okay, we have a number of interesting questions from Andrew here. I'm going to ask a couple of them. Hopefully, if we can't get to all of them, feel free to contact us afterward. Which wild tomato species provide the most interesting new flavor profiles that could be interesting to introgress?
Oh my, that's a very interesting question. Um, there are, many of these wild species have the same volatiles in that are found in cultivated tomato, at least, say the older, uh, not the more modern types, but um, they're present in different ratios, and in some cases they are. I can't remember the compound and the species, but there's a case of one particular compound being like 1,000 times higher in the wild species, and at that level it will cause projectile vomiting when people eat it.
Mm.
So, that's probably not one you're going to want to incorporate at that level. Um, I think there's that's something that could really be explored in depth further, and I don't know what you would get, but there are many different flavors that are potentially would come out of work like that.
Okay. So another question, do you have any recommendations to cross Chilenza to Lycopersicum without embryo rescue?
No,
Okay.
I think just the brute force, maybe that might be about the only way to make that. Otherwise, yeah, it does require embryo rescue. But if you can make thousands and thousands of cross, there might be a chance you would get something. Sometimes it's important to choose the right genotypes. Some genotypes nick better, and for that information, I would go into the literature and see what people have used and see if they have certain, say, cultivated tomato types that seem to work well as in crossing with wide crosses. So that would be my approach to investigating that. One of the things I think about, one of the barriers is some of the wild, the green fruited types, the wild are they have self-incompatibility systems, and that is a real problem because that tends to be inherited as a dominant trait, and so you can get the F1, but then you can't get past that that period or that generation, and so if you can find a self-compatible accession of the wild to start with, that can be important then to be able to continue making crosses.
Okay, we have a comment from Glenn here. He says, woodiness and stress, especially in the late season, will definitely affect taste, more bitterness, which is another measure, and also the species in line. Bacterial wilt-resistant lines taste bitter with solanine and tomatine, and he's breeding tomatoes in Hawaii.
Yeah, I think Glenn's comments are very, very good because they're, tomatoes have these glycoalkaloids, tomatine and some other related ones that can taste very bitter. Um, and, you find them in, particularly in wild species. And there's, there's a form of cultivated or a form of the Solanum-like persicum called variety cerasiforme, which have they're kind of small cherry fruited types. You often find them free living in Mexico, Central America, and South America. Some of these are thought to be a progenitor actually to cultivated tomato. Others may be feral, that they've reversions from cultivated back to a free living form. But some of those may have particularly high tomatine levels in them. And there actually are some cultures who have in South America, I think, who have incorporated these into their cuisine.
Hmm.
So yeah, that's another aspect of breeding tomatoes.
Well, Amber just put in a comment here about the question about wild species. And she said, on the flavor question coming from wild relatives, the research group led by Dr. Mathilde Cos at INRAE in France has carried out several crosses with different wild relatives and looked at flavor profiles. So that might be a good resource to look into. So that's in the chat right there. So maybe if you're interested, you can search for that. That's good to know. Let's see. Another question. Why is pH 5 not yet deployed?
So I think the answer to that is it's in Solanum pimpinellifolium. We are in our third round of crossing to get it into something that has some acceptable fruit size, and we're up to about, say, a paste tomato size so far with this. Um, I don't, what I've seen in my evaluations with the original source of resistance is quite resistant. I don't have as good a feel for how it interacts with the other resistance genes and its overall how well it confers resistance. The other thing, there's another part to this in that the more resistance genes you incorporate into the variety, it reduces your resources to do other things in your breeding program. So it's just tough bringing all these different resistances together. And one of the strategies that's been used pretty widely with the pH genes in tomato and combining pH 2 and pH 3 is you're producing a hybrid, and each of your inbreds will be homozygous for one of the genes. So one inbred's pH 2 and the other's pH 3. When you cross them, you have a heterozygote for pH 2 and pH 3. And that confers resistance, maybe not at the same level you would get if they were both homozygous in that background, but it's still enough to be very useful. And so you don't have to go to the work of creating inbreds that are double homozygotes for the pH genes. And that may be the case with adding pH 5. It's going to, be more work, and breeders may ask the question, is it really worth doing that? Are we doing good enough with pH 2 and pH 3? My answer to that would be that these genes, there are new races of Phytophthora that are evolving, and they can overcome the resistances in these other genes. So just stacking these genes together is going to be the best long-term strategy to having stable resistance to late blight.
Okay, yeah.
Along the same lines, just quickly on the Septoria markers, Ambar's work showed that for one of those, you essentially can get by with a heterozygote. For the other one, it behaves additively, and so each additional dose of that resistance allele gives you more resistance. And so having both homozygous is your strongest resistance, but you can also use one that's homozygous and one that's heterozygous. And that's also in the paper that I linked in the chat. And so you do have to kind of test it to see whether heterozygous is acceptable. But if it is, then a hybrid strategy can work, at least in the short term. And then that gives you time to develop those inbred lines without having to wait for that to get something commercially viable.
Okay, so Jared very helpfully put in some links to some of the papers that relate to Ambar's comment in the chat there. So thanks for that. And yeah, so check the chat for some good references. I'll try to link those on the eOrganic page with the recording. Okay, so here's another question from Roland here. Do you have markers for pH 2 and pH 3 that are in the gene and there will be no recombination?
I think the answer to that is no. That the markers that we've been using are tightly linked, but there is the potential for recombination, and that necessitates the field testing in the very end to validate that you did transfer the alleles that you thought you were transferring. I think it might be possible, with this gene sequences now that we have to go in and identify SNPs that are within the gene, and then to be able to make Caspar markers or something of that sort, CAPS markers that you could use for selection.
Okay.
Another question in the chat was similar to this, and some of these disease resistance alleles come in largely from wild relatives, so that block may not recombine as much, but eventually it will. And so, as Jim said, it's something to be aware of. I think a point that Jared made is that you can't use this to replace field testing, but hopefully the markers allow you to put plants that are more likely to be resistant in the field, so that in your limited testing space, you can focus on all of the traits, and you're more likely to have kind of enriched that group of lines that you're testing for the disease resistant lines, so that your field selection is more efficient. But it can't replace the field selection because things do happen. The markers don't always work, and you really need to look at all the traits together in the field to get a good sense of how things will do.
Okay, we have time for a final question here. Have you found resistance genes that have a negative correlation with taste?
No, we haven't found a problem there, but that's probably because we're using Pimpinella folium, and it's pretty close to cultivated tomato. Don't know if the others have, Julie, whether you've had any experience like that.
No, I do think so. You can find these alleles deployed in backgrounds that are not great flavor. And sometimes if a background has been selected for lots and lots of disease resistances pyramided on each other, you get into that numbers game where it's hard to also select for really good flavor, which is more of a quantitative trait. Individually, I do not think we have seen a specific difference in flavor when we're looking at these septoria resistance lines. It's not to say that we couldn't detect that if we had an enormous population and like we're able to do really sophisticated kind of statistics on the flavor data and the resistance data. But I don't think that a consumer would notice that level of difference. They haven't really been deployed commercially that much yet, though, so I think we'll see if people do notice anything when we see them on a wider scale.
Related to the yield part of that question, I guess, Jim, I see in your table in terms of deleterious linkages, I see it mentioned maybe that the I2 locus for Fusarium resistance showed occasional yield drag.
Mm-hmm. Yeah, there are several cases there that relate to yield and fruit size, things like that. But in the literature, I really didn't find anything on linkages between flavor and deleterious effects of incorporating resistance genes from wild species.
Okay, I think we are out of time. But I'd like to thank everyone for all your questions, and the recording should be available within the week. Link to the website that you see on the screen, the TOMI website, where you can find more information about that project. So thanks, everyone, and have a great rest of the day. Thank you, Jim, Julie, and Jared.
All right, thank you.
Thank you, yes.